Nerve impulses enter through the dendrite and travel through the axon. A dendrite is a short branched extension of a nerve cell, where nerve impulses are received and transmitted. Axons are a part of the nerve cell where impulses are transmitted to other cells. Voltage-gated channels open and close depending on the electrical charge surrounding them. When a neuron is at rest, a charge difference is maintained between the inside and outside. Resting potential is the charge difference in the interior and exterior of the cell. Resting potential is maintained using sodium and potassium channels and sodium and potassium pumps. Resting potential is at a charge of negative 70 millivolts.
First, sodium is on the outside of the cell, and potassium is on the inside of the cell. Potassium channels and sodium channels open. Potassium travels to the outside of the cell and back into the cell through the potassium channel. Sodium travels into the cell and out of the cell using the sodium channel. The sodium-potassium pump keeps up with the sodium flowing in and the potassium flowing out. 3 sodium are pumped out of the cell and 2 potassium are pumped in. This produces a little bit of ATP.
A nerve impulse starts when a stimulus disrupts the plasma membrane. When the charge reaches -5, impulses are transferred to the other cell.
Depolarization near one local area in a membrane causes nearby voltage-gated sodium channels to open, and the depolarization process is transferred across the membrane. The moving depolarization is called action potential. Action potential occurs when the membrane potential is at 30 millivolts. Then, changes occur to restore the potential back to the resting potential, where the membrane potential is at a charge of negative 70 millivolts. This occurs when voltage-gated sodium channels close and the voltage-gated potassium channels open. Potassium ions flow out of the cell and the membrane goes through repolarization. The inside of the cell becomes negative and the outside becomes positive.
Because of repolarization, an undershoot occurs. An undershoot is the opposite of depolarization, and the membrane potential is less than negative 70 millivolts. This is called the refractory period when the undershoot occurs. The refractory period is the period when undershoot occurs, and hyperpolarization must occur to restore the membrane potential to negative 70 millivolts. Another stimulus causes hyperpolarization to occur. Hyperpolarization is the opposite of depolarization. It occurs after the undershoot, and it leads the membrane potential to be restored back to the charge of negative 70 millivolts.
Each local region on the membrane are depolarized and repolarized in a sequence. As sodium ions flow into the cell through the voltage-gated sodium channel, they next repel because they are like charges. The movement of sodium to the next region causes that region to be depolarized. The original region is repolarized, has an undershoot, goes through hyperpolarization, and returns to the resting membrane potential with a charge of negative 70 millivolts. This whole process is solitary conduction. Solitary conduction is in the propagation of action potential along the axon from one node to the next. The voltage-gated channels are separated by myelin.
There are synaptic vesicles in the synapses. The synaptic vesicles, also known as neurotransmitter vesicles, contain neurotransmitters that allow action potential to occur. Neurotransmitters are released from the synaptic vesicles and travel through the synaptic cleft. The synaptic cleft is a space between the axon terminal and the dendrite. There are chemically graded channels located in the postsynaptic membrane. The postsynaptic membrane is the membrane that contains chemically graded channels that receives neurotransmitters from the synapse, and it replies by undergoing a change in the membrane potential. The 2 types of neurotransmitters are excitatory and inhibitory. Both neurotransmitters bind to the chemically gated channels located on the postsynaptic membrane. Excitatory neurotransmitters cause the membrane potential to increase. Excitatory neurotransmitters bind to the chemically graded channels and cause them to open, and there is an inflow of sodium into the cell. Inhibitory neurotransmitters decrease the membrane potential. They bind to the chemically graded channels, which causes the channel to open. Potassium travels out of the cell, or chloride travels into the cell. When the membrane potential reaches negative 5 millivolts, action potential starts.